We are now in chapter 19, the first part of a two-lecture series on the cardiovascular system. We will be discussing the vascular system. And today we're going to be doing the anatomy of and the structures involved in forming our blood vessels. Let's take a look at the relationship of blood vessels to each other and to the lymphatic vessels. You remember that we discussed the lymphatic system. The lymphatic vessels are actually emptying into the subclavian veins. They pick up um, tissue fluid or excess tissue fluid, and eventually they will be filtering it right through the lymph nodes. And then when they're ready and they get right through, go back into circulation, they empty the lymph back into right through the subclavian vein. So the blood vessels here deliver, it's actually the delivery system of the dynamic structures that brings and that begins rather and ends at the heart. They work with the lymphatic system to circulate fluids. The arteries carry blood away from the heart. Meanwhile, your veins carry blood towards the heart. And in between, there's actually minute blood vessels, which we call capillaries, that would connect the small or the smallest of the arteries and the smallest of the veins together. And they are the ones that are also in direct contact with the tissue cells. All vessels consist of pretty much like a lumen. This is actually the lumen. That's the lumen. This is the central blood containing space surrounded by a wall. The walls of all vessels except the capillaries have three layers or tunics. You have the tunica intima, and that's your tunica intima right there, the tunica media or the middle layer, and the tunica externa or the external layer. Meanwhile, your capillaries, and this is your capillary right here, are all single layered endothelium with sparse basal lamina. Let's do these layers. The tunica intima, which is actually the innermost layer, that is in intimate contact with the blood, and that's the reason why you call it tunica intima, is also known as the endothelium of the blood vessel, and it's made up of a simple squamous epithelium that lines the lumen of all blood vessels. It is continuous with the endocardium and has a slick, very slick, they call it non-wettable surface that reduces friction. And there is such a thing also as a subendothelial layer under it, which is I cannot move my thing, which is really a connective tissue, basement membrane, that is found only in larger vessels, that is 1 millimeter thick. Now, the tunica media is a middle layer. It's composed mostly of smooth muscles and sheets of elastin. It is innervated by sympathetic vasomotor nerves controlling vasoconstriction, for one thing. This is actually the muscle action to decrease the lumen diameter, and also vasodilation, which is the muscle action to increase the lumen diameter. This is the bulkiest so far. The media, the middle layer, is the bulkiest layer and is responsible for maintaining blood flow and blood pressure. The tunica externa, which is the outermost layer, is also known as tunica adventitia. It is composed mostly of loose collagen fibers that protect and reinforce the wall and anchor it to surrounding structures. It is infiltrated with nerve fibers, lymphatic vessels, and some large veins that contain elastic fibers in this layer. They are being infiltrated also by large blood vessels, and we call them the vasa vasorum, the blood vessels that infiltrate the big vessels. They need to be fed also. If they are big vessels, they're big organs also, and they need their own vascularization. This is a vasa vasorum is a system of tiny blood vessels that is found intertwined in the muscles and in the layers of the big vessels, and they serve to nourish the mostly the outermost external layer that is not in contact with the blood. Let's take a look at the arteries. There are different types of arteries depending on how thick they are and depending on their function. The elastic arteries, for one, okay, the first one, well, we have three that I can show you today. You have your elastic artery right there in the middle is your elastic artery. Then you have your muscular artery. Then you have your arterial. Let's take a look at how different they are from each other. The elastic arteries are thick walled with large, low resistance lumen or the middle part. The aorta and its major branches are also called conducting arteries. Although they are really very elastic just by themselves, they are also called conducting arteries because they conduct the blood from the heart to the medium-sized vessel. Elastin is found in all of the three layers. Mostly in the tunica media, you have a good amount there, and they contain also substantial smooth muscle. But these, uh, the but the arterioles are inactive. These particular arteries are inactive in vasoconstriction. So your aorta and the big arteries are not doing any vasoconstriction. They only act as pressure reservoir that expand and recoil as the blood is ejected from the heart. It allows for continuous blood flow downstream between the heartbeats. So the elastic arteries has a lot of elastic component in them, elastic connective tissue, because their function will be to receive the bulk of the pressure of the blood coming from the pumping heart, and they need to be able to expand and then recoil whenever there's no pressure or as there's less of pressure. So your arteries are actually throbbing. They are actually like moving. They expand and then they recoil, they expand and then they recoil. Pretty much, this is what we feel whenever there are arteries that are very close to the skin and we feel them and we call them the pulse. Let's now go to the second, and which is your muscular arteries. They are also known as the distributing arteries because they deliver blood to the body organs. Their diameters range from your pinky finger size to pretty much like the pencil lead size. So they account for most of the named arteries that we are able to name in the human body. They have the thickest middle layer with more smooth muscle, but they have less of the elastic tissue. Now, this is sandwiched between elastic membranes and is very active. This particular type of artery, the medium sized artery, the muscular artery, is very active in vasoconstriction. Then we have the smallest of all the arteries, which we call the arterial. And the larger arterial, though, contain three layers, while the smaller ones are mostly single layer of smooth muscle surrounding endothelial cells. These control the flow into the capillary beds via vasodilation and vasoconstriction of smooth muscle. They're also called the resistance arteries. They're arterioles because they change in diameter and would change resistance to blood flow as well. These lead to the capillary beds. The arterioles would lead to the capillary bed. Your capillaries. Your capillaries are microscopic vessels. The diameters are so small that only a single red blood cell can pass through at a time. The walls are pretty much like just thin tunica intima, which is like the endothelium in the smallest vessel. One cell forms an entire, one cell only forms an entire circumference. And there's also like the specialized um, cell, which you call pericytes. And here you could see the pericytes um, in this particular capillary. The pericytes are spider-shaped stem cells that help stabilize capillary walls. They control permeability and they play a role in vascular or in the vascular repair. Now, they supply almost every cell except for the cartilage, the um, the capillary, because the cartilage are supposed to be avascular, so there is actually no blood supply. And for anything that would have blood supply, the capillaries in the capillaries are actually there. So pretty much the ones that are avascular, like your cartilage, your epithelium, the cornea, the lens of the eyes, they do not have any capillaries. So what's the function of this capillary? Well, they are the ones that are giving gas and nutrients. They are also withdrawing waste, and hormones actually move through them and into the tissues. So pretty much like between whatever needs to be exchanged between the blood and the interstitial fluid, pretty much is the capillary does it. So there are three types of capillaries. All capillary cells are joined by tight junctions with gaps they call the intercellular clefts. They allow passage of fluids and small solids. That's a special note that I have to pretty much like mention to you before we start. And there are three types of capillary. You have the continuous capillary, this top capillary here, and then you have your fenestrated capillary, and then you have your ugly looking sinusoidal capillary. Let me explain to you the difference structurally between these three. I will explain them individually. And here you have your continuous capillary. These are abundant in the skin, the muscles, the lungs, and the central nervous system. They are often associated with pericytes. The continuous capillaries of the brain are unique because they form the blood-brain barrier and is totally enclosed with tight junctions. They do not have intercellular clefts. The continuous capillary. That's why you call them continuous capillaries. Then you have the fenestrated capillaries. Take note that the fenestrated capillaries have big fenestrations, and you could see the holes right there. Those are the pores or the fenestrations. They are found in areas involved in active filtration, such as the kidney, or the absorb, or the, in areas where you have active absorption, the intestines, or in areas where you have an active secretion of endocrine hormones. Their endothelial cells contain something that looks like Swiss cheese like pores, like, and we call them fenestrations. These would allow for increased permeability through the through the capillary. Fenestrations are usually covered with thin glycoprotein diaphragm. And let's take a look at the last one, last capillary, which is your, I told you, the ugly looking sinusoidal capillary. Why do we have such capillary that looks like it's dilapidated, that looks like it's already falling apart? Well, the sinusoidal capillaries have fewer tight junctions, and even the basement membrane is incomplete, and they are usually fenestrated with larger intercellular cleft. They're, well, just like what I said, the basement membrane is incomplete, and they have larger lumens. See this? They usually have bigger openings within inside them, and they are found only in specialized organs such as your liver, the bone marrow, the adrenal medulla. The blood flow in this particular type of capillary is very sluggish because the things that actually go in them are very thick, which allows for the reason for the sluggish movement is it allows time for modification of large molecules and blood cells that pass between the blood and the tissue. They contain a lot of macrophages in the lining to capture and destroy foreign invaders. So, the capillary bed. So all of these three capillaries, they are found in different organs in the body. And we're now going to get into the capillary bed. We're going to pretty much like um, discuss a standard capillary bed where you can find in a skin, okay, and it is not it does not have any special arrangement like the capillary bed of the liver or something. So this is just an interwoven network of capillary between the arterial and the venules. So it is showing how the venules and the venules, I'm pointing to the wrong part, and the arterioles are actually interconnected through the capillary bed. They form what you call a microcirculation, like the flow of blood through the bed from the arterial into the capillary back into the venule. Now, the terminal arterial, this is your terminal artery right here, branch off. It's actually an arterial branch that would further branch into 10 or 20 capillaries. So you could see the arterial here, it branches off into 10 or 20 exchange vessels. And these are actually the ones which you call the capillary bed. There's going to be exchange of gases, nutrients, and wastes from the surrounding tissue, and it takes place right here in the capillary bed. The capillary would then drain into what you call a post-capillary venule, and the blood flow through the bed is controlled by the diameter of the terminal arteriole, right here, that's your terminal arteriole, and the upstream arterioles. The local chemical conditions and arterial vasomotor nerves would regulate the amount of blood entering the capillary bed. And the arterial and terminal arteriole are dilated when blood is actually needed. So they're dilated and blood flows through them freely. They constrict. When they constrict, they shunt the blood away from the bed. So when they constrict, when the terminal arterial, okay, I'm right here at the bottom one, when the terminal arterial constrict, they will shunt away the blood from the capillary bed itself when blood is not needed in that particular section. So there is another type of capillary flow within the capillaries, and this is actually the thoroughfare channel. This is what you call the thoroughfare channel. Now, first of all, let me emphasize that the capillaries are found in the serous membrane. Capillaries that are found in the serous membranes, most especially in intestinal mesenteries, have additional features that form a special arrangement of capillaries. The vascular shunt. The vascular shunt is actually made up of the metarterial and the thoroughfare channel. The vascular shunt is a channel that directly connects the arterial with the venule. It bypasses the through capillaries. These are your through capillaries. Then in the middle, you have what you call the vascular shunt. When the through capillaries are closed, or they are actually like when In this particular case, they have what you call the precapillary sphincters. These precapillary sphincters will control the opening and closing of the capillaries, and that will control also the flow of blood in it. When the precapillary sphincters are closed and the blood is not allowed to pretty much like flow into the into the capillary bed, blood is not going to be prevented from flowing directly into the veins, so that the veins can actually pick it up if it needs to be it needs to flow through. It can flow through the thoroughfare channel, and that is a direct channel that is open and will keep open, and that will allow blood to flow from the arterial into the venule. Now, these precapillary sphincters is actually a cuff of smooth muscle. They're made up of smooth muscle surrounding each true capillary branches of a metarterial thoroughfare channel. It acts as a valve. They do act as a valve regulating blood flow into the capillary bed, and they are controlled by chemicals, and they are not innervated. So we're back to the generalized structure of your big blood vessels, or the blood vessel itself. We're now going to get into the veins. The veins carry blood towards the heart. Their formation begins when the capillary bed, from the capillary bed right there, okay, from the capillary bed, unite in the postcapillary venules and merge into larger and larger veins. The venule capillaries unite to form the postcapillary venules. They consist of endothelium and a few parasites. I don't think they are showing the parasites here. They are very porous. They allow fluids and white blood cells into the tissues, and the larger venules have one or two layers of smooth muscle cells and are formed when the venules converge. They all have layers, they all have tunics, but the tunics or the layers of the vein have thinner walls with large lumens compared with the corresponding arteries. The corresponding arteries are thick walled. If you're going to pretty much like think of what you can actually compare the arteries with, you could compare them with your garden hose that is not collapsing and it stays like round even without water in it. Rarely, I think in the old days when I was very young, I remember we used to have a garden hose that's actually collapsing. When it doesn't have any water, it collapses. But then it my mother doesn't did not anymore buy any one of those kind because they broke. They um, pretty much they did not last long, but the veins would probably be just like it. It's actually like collapsible. The brains are collapsible, but our veins are our connected would have um, would not be uh, like that particular garden hose where it breaks pretty much like very very easily. Now, the tunica or the layer of the vein is very thin, but the externa is actually very thick. They contain collagen fibers and elastic networks. That's why they're still very good blood vessels. They have large lumen and thin walls that make them good storage vessels. They are known to be the capacitance vessels, or AKA blood reservoir, because they can contain up to 65% of the blood supply. Blood pressure in the veins are much, much lower than in arteries. So there are a lot of adaptations in the body that is needed, or in the vein itself that is needed to make sure that the veins would return back to the heart and would not be flowing back to your legs. The large diameter veins offer very little resistance, and some of its adaptations will be the valves, and they are really very important. They prevent the backflow of blood. They are most abundant in the veins of the limbs. You don't want your blood to be pooling right there on your legs. We also have venous sinuses. These are flattened veins with extremely thin walls. And examples of this would be your coronary sinus of the heart and the dural sinuses of the brain. And you will be encountering them when you work on the blood vessels in the laboratory. And this is the histology, which is pretty much what you need to remember. You are going to have to know the difference histologically between an artery. This is actually your artery. Take note that the artery is most of the is actually always round or oval. It doesn't collapse, and a vein usually would be collapsing. A vein is thin-walled, and your arteries have are thick-walled. And those dark areas are actually the elastic fibers. And you could see how much elastic fibers a an artery would have, and very thin layer of elastic fibers for the. I mean, you can see the very thick elastic layer of an artery. I take that back, and the thin elastic layer of a vein. And here's actually one of the fun fact about the vein. Uh, Pretty much a relatively high proportion of blood volume throughout our cardiovascular system is being kept by the vein, and approximately about 60%, and that's really very big, bigger than half of your blood volume are actually in your veins. And you could take a look at it. The systemic arterioles carry only 15% of it. The pulmonary blood carries 12% of our blood. A heart carries 8%. Capillaries have 5%. And the rest of the 60% is carried by the veins. And that's the reason why they're called the blood reservoir or the capacitance vessels. Here's a homeostatic imbalance that pretty much annoys a lot of people, most especially women. And that's actually the varicose veins because they are very unsightly. It is when your veins, most especially on the legs or in your limbs or in your hands, would be dilated. And for the most part, they become very annoyingly painful due to incompetent or leaky valves. The factors that contribute to the development of this, most especially through as we age, is actually brought about by genetics. We inherited the uh, our predisposition to having varicose veins is actually inherited. Vascular anastomosis, a term that connotes interconnections of blood vessels. This is where your artery, two parallel arteries can actually meet at some point, and that's what you call an arterial anastomosis. And right here, this thing looks like the heart. It's because this is a coronary artery of the heart. The anterior, the anterior coronary artery and the posterior coronary artery, anterior interventricular, posterior interventricular, actually would anastomose right there at the apex of the heart. The good thing with this is that if you have a blockade, if you develop a blockade right here, your heart your heart muscle will not immediately die because you have collateral. Anastomosis is very good. It allows collateral circulation on the other side. So this is now on the other side, on the other hand, this part here is actually an arteriovenous anastomosis. It's actually a connection between an arterial and a venual. Now, the capillary bed is actually a typical arteriovenous anastomosis already. And you've seen how an arterial actually have connected into a veneal through through the capillaries. But other than that, you have a veneal and arterial, which is actually a arteriovenous anastomosis. Like your thoroughfare channel is an arteriovenous anastomosis. Then you have your venous anastomosis, which is actually the most anastomosis, the most visible of all of the anastomosis and branching of all the three different types of blood vessel. The veins actually branch out really dramatically. Okay, that's the end of this particular part. We are now going to get into part two.